Okay, so we're recording now, guys, just to let you know. All right, so thanks. So in pathology, change my color here. In pathology, we're actually studying the disease itself, and etiology is studying the cause of a disease. So you may hear the term etiological agent, etiologic agent, and etiologic agent is the same thing as a causative agent. And pathology is the actual disease itself. So what is this disease doing inside of the body? What kind of damage is it causing? And that's also referred to as pathogenesis. So a pathologist, right? A pathologist is somebody who observes and identifies disease tissue. So you spend about 10 to 12 years in college, in graduate school, up through graduate school, becoming a medical doctor, a veterinarian, or a dentist. And then you can go back and spend another 4 years becoming a pathologist. So when you go to med school, you study healthy tissue. What is healthy tissue supposed to look like? And you, of course, study signs and symptoms of disease and those sorts of things. But a pathologist is somebody who actually specializes in the microscopy and such of disease tissue. An infection, we know, is when we have pathogens that are actually growing and colonizing within the body, and a disease is, of course, an abnormal state. Now, we always have to talk about, when we talk about disease and principles of disease, we always talk about our own normal flora. Remember, normal flora is kind of populated microorganisms that are found on our skin and within our body. They are a part of us physically. But we also sometimes pick up what are called transient microbiota. And so transient organisms might be available for a couple of days or weeks. Some can stick around for months, but most of the time we think of transient microbiota as those organisms we pick up when we touch a doorknob or a handrail or an elevator button, those sorts of things. Everybody's all about washing hands right now. So in washing your hands, you end up physically removing those transient microbiota. Your normal microbiota would stay behind. Your normal microbiota tend to be growing in your fingerprints. They're kind of more firmly attached to you, such as Micrococcus luteus, Staph epidermidis, and Staph aureus. So normal microbiota are permanent colonizers of us. In symbiosis, that's that relationship that we have between our normal microbiota and ourselves. I help you, I give you a place to live, you help me, you protect me from transients and provide me with some sort of benefit. So just to review these symbiotic relationships, remember commensalism, where one organism is benefited but the other is kind of unaffected. That's what we sometimes think of in our skin flora. In mutualism, both organisms are going to benefit. So that's what the kind of relationship we usually have with our gut bacteria. Of course, parasitism is where one is benefited at the expense of another. And then we can see that, of course, with some of the protists and those types of organisms. Our normal microbiota can become parasitic if they're given the opportunity. So we call these opportunistic pathogens. You give them the opportunity and they are going to cause infection. Now, in how our microorganisms assist us or how they help us, our normal flora, we did discuss this a little bit in immunology, so I just want to kind of review it real quick. Our normal microbiota can help us by occupying niches. So they will occupy areas in the, say, in the gastrointestinal tract. And they will produce these bacteriocins. They'll produce these small little proteins that will inhibit other bacteria from occupying those areas. So you can kind of think of it as kind of no vacancy or no room at the inn. They can change the pH of an environment in order to either survive in that environment or to perhaps prevent other microorganisms from growing in it. And they can outcompete transient microorganisms for food. They can produce compounds that force, say, they can force something like a Clostridium to go into an endospore form, so it's dormant. So it can be present, but it's going to be dormant. We talked about Koch's postulates in the past as well. I just want to review it real quick. Remember, in Koch's postulates, when an organism here, Koch was looking at Streptococcus. So once an organism is infected, right? So we isolate microorganisms from a dead organism. We take a look at one of the colonies under the microscope and we can identify the microorganism. We then take bacteria from that culture and we inject it into a living healthy organism. If that healthy organism produces the same disease, then we isolate microorganisms from that organism. We look at it under the microscope, and of course, it should be identical to the original microorganisms that we identified. And this is how, this is the basics, or I'm sorry, the basis for determining what organism causes disease. So this is the type of thing that was actually done with viruses, with the COVID. So what they did in China was they recognized that this was a viral infection, and they used PCR. And they started sequencing the genome of this virus. They discovered virus in tissue, and this would be a modified Koch's postulate. So they identified virus in tissue. It was observed under the microscope, under an electron microscope. They knew that it was an upper respiratory, and they probably ran PCR with multiple primers for multiple families of bacteria. They finally found it was a coronavirus. Once they knew that it was a coronavirus, they then started comparing it. So they started sequencing the genome. And once they sequenced the genome, they found that it was closely related to SARS and MERS. So they were using Koch's postulates, they just weren't reinfecting people with it. What they were doing is they were finding the same virus in multiple patients. So they were looking at all of these multiple patients. They were using PCR to compare the genome, and they finally realized that it was a coronavirus, but it was a coronavirus that is not on record, that no one has ever seen before. It is very, very, very closely related to SARS and MERS. Genetically, there's not a lot of differences, but there are just enough that it's considered a novel virus. The biggest difference in it is SARS was actually a much deadlier virus, had about a 10% mortality rate, which is very high. SARS is much deadlier. But the biggest thing is SARS, around the world, there were just around 8,500 cases, approximately. That number is approximate. SARS was not as easily transmissible. That was the big difference. Now, don't get me wrong, COVID has about a 2 to 3% mortality rate right now. It's not really truly determined what that mortality rate is. But its transmission, which is called an R rate or an R value, its transmission is greater than 1, which means that we're on the upswing of transmission. We'll go over that a little bit later. So this is a new virus, and it has a pretty decent, 2 to 3% is a pretty decent mortality rate, and they used the basics for Koch's postulates to identify this particular virus. Now, there are sometimes exceptions. We talked about exceptions in terms of like viruses. We talked about modifying Koch's postulates to identify toxins and other pathogens. But there are sometimes a few exceptions to Koch's postulates in terms of signs and symptoms. Sometimes we have, of course, unique cultural requirements. So this could be viruses. If we need to grow the viruses, we have to do this in cell culture. Bacteria we can grow in regular media, but viruses, they require cells. Same thing with like rickettsial diseases. They are what are called obligate intracellular pathogens. So we have to have cells in culture in order to grow these organisms to study them. The other are unique signs and symptoms, and the two examples of this are diphtheria and tetanus. In diphtheria, if you have a young child that comes in and they have kind of this kind of seal bark type of cough, They're usually not able to talk very well. Breathing can be a little bit difficult, but not because the lungs are filled up, but because their passage, their airway is obstructed. They get this kind of membrane here over their tonsils and into the vocal cords, and it causes this narrow opening. So they have like a whistling sound when they're trying to breathe, and they'll cough. And that cough will sound like a seal bark. Over here is tetanus. This is an actual tetanus patient. What happens in tetanus and what makes it unique is that most of the time when we see paralysis, we think of what's called flaccid paralysis, okay, where somebody cannot contract their muscles. But in tetanus, we have the actual opposite. This is called rigid paralysis. And rigid paralysis usually occurs in one of two situations. The first and most common would be due to head injury, where the muscles are receiving mixed signals from the brain, and it causes constant muscle contraction. The second is through tetanus, and this is why tetanus is called lockjaw. It's sometimes referred to, that's the common name for it. Now, what happens in tetanus is acetylcholine receptors. Acetylcholine receptors are turned on. And these are the receptors that our muscle cells use to receive signals from our brain. Excuse me. And so the tetanus toxin binds to these acetylcholine receptors and activates them permanently so that all muscles contract at the same time. And you see this kind of posturing that occurs here. I know this is a rough picture to look at, but you see this posturing that occurs here. The hands are pulled in, the fingers are tucked under, the arms are folded up. And this is because although both the bicep here and the tricep under here are both contracting, the bicep is the stronger muscle. So this muscle in the back here is trying to contract. It is contracted, but the bicep is stronger. So it's going to pull the arm inwards. It's referred to as lockjaw because the jaw itself is considered one of the strongest muscles in the body because we use it the most. And so when this occurs in the mouth, the teeth are clenched shut and the person cannot open their mouth. So they can't eat, they can't drink. It's a very painful disease. That's a very late stage of tetanus. All right, so when we classify infectious disease, we have to talk about symptoms and signs, and then together the combination of these two things are going to give us what we refer to as our disease or syndrome. So the differences here is that a symptom is something that a patient's going to feel, but we can't measure it. Right? So these are questions, these are when you're doing triage in the ER and you're saying on a scale of 1 to 10, you know, how much pain are you in? We don't have a way to scientifically measure pain, so to speak. So we have to rely on the patient. So we try to use a fairly unbiased method in a scale. So we ask them on a scale of 1 to 10, how painful is that? How anxious are you? How dizzy are you? A sign is something we can measure. So for symptoms, these could be things like nausea. How do you measure someone's nausea? Fatigue, dizziness, pain, I don't know, think of another one. Then we have signs. These would be temperature, respiration rate, heart rate, urine output, those sorts of things. So things, there you go, BP, you can measure BP. So the signs are things that we can actually measure. We have a value for them. We have some kind of scientific way of determining what that sign is. A symptom is something we have to rely on the patient. And it's important as healthcare providers that you remember that when you ask, like on a scale of 1 to 10, how painful is it? If that person tells you it's a 9, to them, it is a 9. Even if you're looking at it and going, okay, you have a hangnail or you have an ingrown toenail. To them, it's a 9. So pain to them, to a patient. Pain is felt differently by people. I can tell you that men tend to have a higher scale for pain than women. And what I mean by that is when you ask on a scale of 1 to 10, what hurts as a 9 for a man is probably about a 7 for most women. Women do have in large numbers, it's been shown that women have a higher pain tolerance, and that's believed because they have evolved to bear children. So women do tend to have a higher pain tolerance. So when we get into epidemiology, now we start looking at infectious disease based on how that disease behaves not only in a single host, but within a population as well. So we have diseases that are considered communicable. Like with COVID right now, we are looking at a communicable disease. Really, it's been upgraded to contagious, and we'll talk about that in a minute. And a communicable disease, this is one that is spread directly from one host to another. So this can be through direct contact, such as we have here, right? So wash your hands, careful when you're kissing. So we can have direct contact, but it can also be through indirect contact, which is through touching surfaces and, of course, inhalation. So respiratory droplet type transmission, which is what we see initially with our COVID that we're looking at right now. But COVID has become more of a contagious disease. And this is why when we have outbreaks of a cold or the flu, these are communicable diseases. And we don't have quarantine, but here we have this really highly contagious disease. This disease spreads very, very easily. Flu is a contagious disease. Strep throat is a contagious disease. And of course, now we'll have to add to this COVID. So COVID is also considered, or COVID-19, let me be specific, is a very contagious disease. There's concern because it can survive on surfaces for a long period of time. It is a droplet transmission. I heard talk of it being airborne. I'm not sure of that yet, so I don't want to go down that road. But it is respiratory transmission, so droplet transmission, coughing, sneezing, talking, anytime, any type of droplet is being released by someone with COVID, there will be virus within that fluid. A non-communicable disease is one that is not transmitted from host to host. These are usually occurring in reservoirs of disease, such as bodies of water. Tetanus is considered non-communicable. If you treat a patient with tetanus, you're not going to contract tetanus from that person. Tetanus only causes disease when it's introduced into the body, but if we wanted to, we could go outside and we could isolate the tetanus bacteria from soil. It's found in soil everywhere, but it is not until this organism actually starts growing within the body, is introduced into the human body, or into a mammal, warm-blooded mammal, does it actually start to produce disease. And the reason for this is the tetanus organism itself is not the problem, it's the tetanus toxin. And so when you get vaccinated against tetanus, you get an antitoxin. So you actually are being introduced to a version of the toxin in order to produce antibodies against the toxin. The organism itself is not the problem. Malaria is a non-communicable disease, correct. When you treat someone with malaria, you're not going to get malaria. It's not transmitted host to host, it's transmitted through a vector. Same thing with Chagas or trypanosomes, any of those vector-borne diseases, they are non-communicable in the sense that they are not transmitted from one human to another. They are vector-borne diseases, which is a whole different class. So how often does disease occur? And you may have heard some of these terms being thrown around in the news. So when we talk about occurrence of disease, we have first off, we have incidence and prevalence. So incidence is a fraction. It's a value that's telling us how many people contract a disease during a given time. So an example of this would be a statement from a report on HIV a few years ago, which is in 2009, 48,600 new cases of HIV are reported. And what this is telling you is how many new cases. Now, right now, just to bring it back to our COVID situation, right now in the COVID situation, they are looking at 5-day intervals. I'm going to show you that in a minute. We're going to go to a website in a second. So right now they're looking at 5-day intervals, and that's what they're calculating this curve on, that transmission curve, or sometimes called an epidemiological curve. And when they're talking about flattening the curve, they're trying to reduce the number, they're trying to reduce the incidence of disease. Now here, normally in epidemiology, we would look at months at a time, or we would look at an entire year, data for an entire year, to come up with what's known as an incidence rate. But right now, because we're in the midst of a pandemic, they're looking at an incidence rate in terms of 5-day intervals. Now, prevalence is the amount of people, or the part of a fraction of a population that has the disease at a given time. And so when they're talking about current cases, they are talking about prevalence. How many people in the population actually have this disease right now? Now, a prevalence rate is not going to be, right now in our COVID situation, the prevalence rate is going to be all over the place because we have new cases being reported, testing is being done, and of course, the more we start testing, the greater that incidence rate is going to get. As we have people starting to recover, we could see our prevalence rate start to kind of even out. So prevalence and incidence right now are very fluid in the current pandemic that we are looking at. We could look at historical data from other pandemics or other outbreaks of disease. I used HIV just because there's a lot of data on it. But in prevalence, how many people actually have the disease at a given time? So right now, today, what is the prevalence of COVID-19? We would have to go and look at one of the reporting agencies to figure that out. Now, a disease that is considered sporadic, endemic, epidemic, or pandemic, right? So we're going to add COVID here, down here. So a sporadic disease is one that pops up in a population. We'll see usually small little outbreaks of it here and there. It shows up and then goes away. One example of these are foot and mouth disease. We see this in kids quite a bit. Another one is called fifth disease. These are usually small little outbreaks here and there in small areas around the country, and they pop up, and then for a year or two we don't see them, and then they pop up somewhere else. A disease that is endemic, it's one that is always present in a population. It's always floating around somewhere. Somebody has a cold. Chickenpox used to be endemic, but now that we have a vaccination against it, it's not really as endemic, and it might actually be downgraded to a sporadic disease. We would see outbreaks of chickenpox in unvaccinated populations. An epidemic is a disease acquired by many hosts in a given area in a short time. At the time that I wrote this, we were under a salmonella outbreak. Flu is considered an epidemic. They always call it the flu epidemic because we see patches of this around the world. COVID started out as an epidemic. It started out as an epidemic in Asia, and of course originally in Wuhan province in China. And then of course we have a pandemic disease, and this is a disease that is worldwide. It's kind of obvious we are in the middle of one right now, which is why it's kind of interesting to teach this lecture to you while we're in the middle of a pandemic. So I want to share with you a site that we can talk a little bit about incidence and prevalence, and we can kind of see it in action, which is really kind of interesting. So Johns Hopkins University is kind of has been the center point here in the United States. They've been reporting the majority of cases, and they've been tracking everything. The Johns Hopkins, it's called the Corona Resource Center. It is through their College of Public Health. And they are considered the #1 premier college of public health in the world. So if you want to go into public health, go to Hopkins. They have the #1 program in the world. Okay, so here we have in their resource center, they have a ton of resources that are really amazing. Anybody can access this. It's totally open to the public. So the first thing we are always interested in looking at is the map, right? So this is what they call their dashboard. And this is the map of diseases. It'll pop up in a second. It takes a minute to load on my machine here. There we go. So we can see here this map. Look here, you can see the United States is quite intense. We have a lot going on there. Here on the left, they have total confirmed cases. So this would be your incidence. Well, this would be prevalence actually right now. So how many people at this given moment have it? Over here on the right, we have the total number of deaths. Now, these are worldwide numbers. This is not just in the United States. This is worldwide. And yesterday, when I looked at this, it was 181 countries or regions. It is now at 183. So 2 additional countries have been added to our country list here. On the right, we have total number of deaths, and we have total recovered. The concern here is that the total recovered number is not growing very quickly. And this is a slow number to increase. The death number is not a slow number to increase. And of course, the confirmed cases, tested cases, is also not a slow number. Down here in the bottom right, this is the curve that everybody wants to flatten. So this is the worldwide curve of how many cases, right? How we got up to this 1.3 million. So that was what, yesterday, on 2 days ago. So 1.3 million on April 4th. April 4th is the last time they recorded that. So that's a really big number. We can look at this in terms of growth. We look at a logarithmic rate. So you can see that we have some exponential growth occurring in the number of cases and the prevalence of this particular disease. So it's growing pretty quickly. And then we have what's called a daily increase. So here they're using a bar graph to show you the increase in the number of cases being reported. And once it hit over here in the United States, look at this in the beginning of March, is when we started seeing it here in the United States. And in the US, the numbers have gone crazy, right? So we've had a huge spike in cases. But there are factors that contribute to this. What do you guys think are some of the factors that contribute to these big rates of increase before we go to some other graphs? Definitely increase the testing. That's a really big one. That's a very big one. What else do you think in addition to increased testing? That's probably the biggest factor, absolutely. Those numbers were much bigger originally than they thought. Not everyone is reporting on the same page. What do you mean? You mean other agencies are getting data that maybe Johns Hopkins is not? There's 3 places that everybody has to report to. Yeah, there are 3 places everybody is supposed to report to. They're all supposed to report to the CDC, mostly countries, yeah. So here in the United States, everybody has to report to the CDC. They have to report to their local health department and their state health department. And then Hopkins accumulates all of the data from CDC and local and state health departments. Yeah. Jonathan, flu season, that's an interesting idea. How do you think flu season is affecting these numbers? Yes, Megan, that would slow down, hopefully. Jonathan, how do you think flu season affects the incidence of COVID-19 right now? The time of year. Yeah, definitely. As far as flu goes, absolutely. Flu season is usually in the winter time. In the northeastern part of the United States, our flu season actually starts around August, September. We start a little earlier, and that's mainly because for us, it's really hot that time of year. So that's when we're always inside in the air conditioning in recirculated air. And so we see a little bit greater of a rate of flu down here in the hotter months, whereas up north, they see it in the colder months when people are indoors a lot. Jalen, that could be. That is why testing is so incredibly important. Absolutely. So they could have presumed cases and they're testing for it and they just don't have those results back. So you would probably see over here, that's why this on the left, it is total confirmed cases. So those are cases that have actually been tested. This would probably be in the 3 million range if we were just looking at people that were symptomatic. And imagine how many people are out there that are asymptomatic. That's a big characteristic of this disease. Many people are out there and are asymptomatic. Now, I want you guys to think about something real quick. When we talked about viruses, does anybody remember when we talked about antigenic shift and antigenic drift? Jalen, yeah, okay. So can anybody tell me what the difference is between drift and shift? Right, so that we have some immunity to it, but not necessarily great good immunity, right? We don't have our immunity is not as efficient. Correct. A shift would be a completely different virus. So in COVID, since COVID is so closely related to SARS and MERS, do you think that's a shift or a drift? I'm going to have to agree with Victoria, and it's a drift. SARS was the shift. So SARS was a coronavirus that just came out of nowhere, okay? Came out of nowhere. That does not mean that COVID is not a novel virus. It is a novel virus, but it's incredibly closely related to SARS. So it's a drift from SARS that is enough of a drift that we don't have any immunity to it. But think about the SARS outbreak. If we go back to the SARS outbreak only affected less than 10,000 people worldwide, and they never were able to create a vaccine against it. So what that means is that if this were SARS instead of COVID, we would be in the same boat, because nobody has immunity against SARS, nobody has immunity against MERS, which is another drift from SARS, and then nobody now has immunity from COVID. And that's why this, when COVID showed up, COVID is so highly transmissible and moves so quickly through a population. That's why we are reacting the way we are with COVID, but we did not with SARS. So COVID is really a drift, but it's a drift from a virus that none of us had immunity to begin with, which essentially means that that there was no, it has the same effect as if it were a total shift. Does that make sense? You guys see that? Yes, no, maybe. Does anybody have any questions so far? No, you guys are doing okay? Okay, so I want to go back here. And the reason I talk, yeah, Victoria, what's up? Oh, that was a oops. Victoria, did you have a question? Yes, it would be just as successful. It is in Africa, but Africa is more rural. The population is very, very spread out. Where do you see the highest concentrations? Take a look at the map. Think about it. Where do you see the highest concentrations of this virus? And there's a couple of factors that have to do with that as well. Right? We see it in areas of high population. So we see it in areas where people are right in highest concentration of population. Exactly. So we see it in really high concentrations where we have people living very close together. We also see it in wealthier countries. Why do we not see as much of it here in Africa or in parts of South America? What do you think? Why would an economic factor be part of it? Lack of testing. Exactly, Megan. They're not getting tested. They don't have the resources to do a lot of testing. In Niger and Congo, these areas, South Africa does because it's a larger city. But in these smaller countries, look at Ethiopia. Ethiopia probably has a lot more cases because they have open markets and those sorts of things. But Ethiopia is a very poor country, and it's under a lot of political stress. So there's not a lot of hospital and healthcare resources. There's probably very little testing going on there. And there's probably not a lockdown or shutdown. So more than likely the numbers in parts of Africa are probably, the true infection rate is probably much, much higher than what is being reported because they don't have testing. That's really a big problem in that area. But look here on the United States. Look, you watch over the next week or two, we're going to start seeing the U.S. start to become entirely red as it begins to move across the U.S., almost like a wave. Of course, where we have these larger cities, let me see if I can't enlarge that. Where's my dashboard here? It's very sensitive when I enlarge. There we go. Let me see if I can move the map over. Okay, there we go. So you can see if we enlarge the map just a little bit, there we go. You can see in the areas where we have really large cities, of course, Miami is becoming a hot spot, New Orleans, and of course, San Francisco, LA is another hot spot. It's hard to see Washington. Let's get that. There we go. That's a little better. So Boston, New York, Philly, which makes a lot of sense, right? People are living very close together. They have mass transit where people are in, like, you know, those subway tubes and buses and those things together. Washington, D.C., of course. Detroit, Chicago. Detroit is becoming a big hot spot. Miami. What happened in New Orleans? Does anybody remember why New Orleans became a hot spot so quickly, almost overnight? Yes, Mardi Gras. They did not cancel any of the Mardi Gras festivities, and Fat Tuesday went on as usual, and all of those people were in New Orleans and partying and crowded and all of that kind of stuff. So all of that happened. That's why they were canceling these big, large events. Now, I want to take you to one other site in this Hopkins board. I'm going to kind of go back here, and we're going to take a look here. This is called the daily update. So has the curve flattened? And so here they're looking at the 10 most affected countries and what's happening over time. I'll make sure you guys can still see this. Yes, great. So here is their 5-day average. This is where they're looking at the outbreak and confirmed cases on a 5-day average. And what we have here is all of the countries all at the same time. Can you take a guess who this is, right? So here's the US. So over 5-day averages, confirmed new cases. So from March 15th, where are we? March 15th, we were down here way below Italy. I'm trying to find us. We were very low, right? So March 15th, we were here. Let's go 2 weeks to March 19th, and we're from 800 cases to 22,000. It's probably a little higher than that's actually up here, 24,000. So that's pretty major spreading. That's a very communicable disease, right? A highly contagious disease. Now we can come down here, and here they give some descriptions. Right, so the line is what's called a 5-day moving average, so kind of the rate of transmission over a 5-day period. And then the bar graph that this is following is the actual data on that day. So here in Italy, they had a big spike right about here, and they had a huge number of cases that were shown there. Spain has also had a major spike, but they seem to be on the downside, so they're starting to reduce the number of cases they're reporting. France has had a huge spike. I'm not sure what happened in France over this period. I don't know if there was something going on in Paris, but they had a huge increase in the number of cases. It could have just been introduction of testing. Here we have the United States, which is actually, they're showing on a downtick, right? So as far as the 5-day average of confirmed cases, there's a bit of a downtick here, but you can see in actual data, it's much higher than that. The UK has also had a major spike in cases, and you can see all of these countries. Theirs isn't until March. But what was going on in China? Why did they have this huge spike here in February? Same reason all of these other countries are seeing these big numbers, and it's because that's about the time that testing was being introduced. Remember, they were much earlier in this than the US and most other countries. So even though we are seeing these big spikes in here, we think, oh my gosh, all these people are getting sick so quickly. Part of that is that they were already sick, and the testing is now catching up to the disease. This disease spreads very, very rapidly, very quickly through populations. And honestly, almost, I would say majority of the world got kind of caught off guard with this. And that's some of the political controversy that's out there now. So these are actual cases where you can go and take a look at 5-day averages and see here they're showing that we're on this little downtick here. We'll see if that lasts as this moves across the country. We can also look at this data in terms of mortality. So in Italy, the mortality rate was at 12%, just over 12% mortality rate. That is really high. Here in the United States, we're at about a 2.9, so somewhere around 3%. This could grow, we'll see. But, you know, the UK is at 10, Spain is at a 9%, and they're now on the downside of their curve. France is at an 8%, they're still on the uptick. So we can start seeing some mortality rates getting kind of rough. One thing to think about in these mortality rates, what are some different factors you think that could affect the mortality rate in different countries? Age, but think bigger than an individual patient, Megan. I'm think population-wise. Victoria, yeah, but Victoria, that would be more towards like incidence and rate of infection. I'm talking about a mortality rate, the number of people dying compared to the number of people with the disease. Jalen, yes, that's a big factor, developed versus underdeveloped or undeveloped countries. Yeah. So what do those developed countries have that the non-developed countries do not? Correct, healthcare systems. Availability of healthcare, absolutely, that makes a difference. What's another thing? What happened in Italy? Italy's not an undeveloped country. Italy's they're, I mean, economically they struggle, but they're not considered an undeveloped country. What do you think happened in Italy? And here, Olga, countries with a high rate of obesity. I would have to look at that. I'm not sure how obesity would play a role, but Megan, they definitely had a lack of ventilators in Italy, but what is going on? What happened here in the US? Why is this so out of control in the US right now? What about social distancing, Jessica? How do you think that affects it? Correct, Megan, absolutely. Lack of it. Yes, Jessica, right. So what we had, not only here in the United States, but in other countries around the world, it's not just the US, France, Italy, Spain, those countries, they were slow on the response. Everybody was. Why do you think we, all of these big countries were slow to respond to disease? That's definitely a factor. We didn't get accurate information. And culture, Megan, correct. Yep. I also think there was not just a delay in information transmission. I don't want to say, well, China hid their numbers, because they really didn't. They just didn't make a huge deal of it. And I know here in the United States, it kind of fell on deaf ears. I can't say that for other countries, but here in the United States, the warnings of that kind of fell on deaf ears, and so we just did not prepare for it. And so we didn't respond fast enough, and this is what happens with pandemics. when you don't respond fast enough. That's not to say that if we had responded more quickly, we would not still be in a lockdown situation. More than likely, we would. Because this disease is so easily transmissible and spreads so quickly, it's definitely something that we would be responding to the way that we are. So let's take a look at some of these this mortality analysis. So here are the number of cases and the fatality rate per country. So here in the US, you can see we're at a 2.9%. That's because of almost 3 people per 100,000 people in our population that are infected with COVID will die. So we're looking at almost a 3% mortality rate. That's a pretty high mortality rate. Let's take a look. What is the flu virus? I had to look that up before class. So I'm just looking it up real quick on another site. Where is flu virus? Everything is corona, so trying to find flu virus data is not easy. Okay, well, this is an interesting article. Let's see if I can't find a graph on here. So I'm going to share a new page with you real quick, or I'm just going to capture it. That's easier. I'm just going to share this with you, and then we'll end for today. Let me get back here. So these are the flu rates and such. Just to kind of go over this before I get out of here. So here are all of the different countries and their mortality rates. Does anybody want to look at any specific country? Is there a country that you're interested in? Maybe that your family is in or something like that? Does anybody want to look at data for a specific country? Jada wants to look at Jamaica. Okay, let's look at Jamaica. Let's see if we can find it on here. Has Jamaica had any deaths that you know of? They haven't had any deaths. They may not be on this list. There it is, Jamaica. Jamaica has, looks like 58 new cases. They've had a couple of deaths, so they're looking at about a 5% mortality rate right now. But that's because their numbers are really low, so that mortality rate could go down. Yeah, 3 deaths, yeah, yep. So their mortality rate could go down. 3 out of 58, that's a 5% mortality rate. So exactly. So if the number of cases increases at a greater rate than the number of deaths, then that mortality rate is going to go down. Does anybody else have a country they want to look at? Singapore and Russia. Okay, so one that's kind of interesting on here right now, I thought was, where did it go? Singapore, right here. Singapore is like the 6th or 7th one down there. Singapore has 1,300 new cases, but only 6 deaths. So their mortality rate is really, really low. So what are they doing in Singapore to keep that mortality rate really low? Right? They have twice as many deaths as Jamaica, but they have, look at how many more cases they have. Right? So they have 1,300 cases. They have a really low mortality rate. This may be a more accurate number because they are doing more testing in Singapore than they are probably doing in Jamaica at this time. So their healthcare system is unique, Victoria. You are correct. They have an interesting healthcare system. So we wanted to also look at Russia and Colombia. So let's take a look at Russia. I need to go back to that page. See if we can't find Russia in here. Russia is very low, if I'm not mistaken. They closed everything down and were like, not here. Their numbers are pretty low, or they're just not reporting them. So Russia has 5,300 cases, 45 deaths, they have a less than 1% mortality rate right now. So there's concern that Russia is not reporting their numbers accurately. I don't know how true that is. I don't see why these health commissions would do that, but you never know. You never know. Colombia was the other one. Here's Colombia right here. Colombia has almost 1,500 cases, 35 deaths, so they have about a 2.4% mortality rate. They're on the same track as the United States. They're just a little behind us in terms of mortality rates. Why do you think the US mortality rate is so high? I mean, we like to use rates because they kind of even out in terms of populations, right? So the population density of a country doesn't make as much difference when we start looking at percentages and rates. We're looking at, you know, per capita type of numbers here. So why do you think the US, Jada, they're testing on deceased patients? Interesting. I never thought of it that way. So we're going back to people that have died and we're saying, did they die on, did they die because of COVID, right? Correct, they're not testing all exposure cases, I agree. Yeah, there's certainly not nearly enough testing going on. Deja, I'm telling you, they're not testing because they don't have enough of them. They don't have enough tests. They're just, and if the person is not showing severe enough symptoms, they're going to send them home. They just don't have enough tests. There's so much confusion in the testing right now. It's crazy. There's about 5 or 6 different ones coming out. Yeah, Megan is saying they're not testing unless it's severe. So they're doing the chest x-ray to see if you're starting to drown. Yeah, they're sending them to testing sites because the hospitals, 104.5 temperature, yeah, they should be, you would think they would be tested, but if they ran the chest x-ray and they don't see anything in there, they're sending them home. Really because they're trying to reserve their hospital beds for the most severe, for people who may need to be intubated or be treated. A high fever is not enough reason to put someone in the hospital, and flu can, yes, and flu can cause that as well. They declined first responders that are moving COVID patients. I know it's crazy. The problem is, like Memorial, for instance, Memorial has their own test. They didn't wait for the FDA, they didn't wait for the CDC. Memorial runs their own test. They have their own laboratory to do it. But that limits them in the number of tests that they can run. So what they have is the testing sites where, like at Hard Rock, I think was one of them, they had some drive-through testing sites that are opening up. Those sites take a sample, they get your information, and those samples are at CB Smith, right? They send those samples to private laboratories. And so private laboratories are doing the kind of the general public testing because they can handle that capacity of testing. They can handle that number of people. And so those tests are going. Now, it can take 2 to 3 weeks to get your result back. That's a big problem right now because even the private laboratories are getting overrun. So they are devoting everything they can to COVID testing, but you got to remember, not every person in the, yes, a big problem. So not every single person in the hospital is in there for COVID. See, these guys still have to run other tests as well. Quest is 2 weeks behind. LabCorp is 2 to 3 weeks behind. Deja, you in the lab? So Memorial is reserving their in-house testing. Oh, you sent the test to the lab. So Memorial is reserving their in-house 24-hour result or 12-hour result testing for those patients that are in the greatest need. When those patients come in and they have fluid in their lungs and they have a high fever and they're multi-symptomatic and they need medical intervention, those are the patients that they're testing really quickly. Yep, non-emergency patients. They have to go to the testing sites, and it's just simply because the hospitals do not have the capacity for it. They just don't, because we were delayed. We were delayed in developing tests. We were delayed in adopting tests. The CDC kind of, I mean, it's just an opinion, but the CDC really kind of dropped the ball on this one when it started getting brought here in terms of testing. And that was part of the reason that it was spreading so quickly, was because the testing criteria was far too narrow. I'm still waiting for that lateral flow device to be introduced. I know that they've Jalen, they're not using their plasma, so to speak. They are looking at developing monoclonal antibodies. That was what they used in the Ebola cases. So when the patients with Ebola, they introduced Ebola antigen into monoclonal B cells. Yeah, Deja, if the person you're talking about is not getting better, I definitely call the 24-hour hotline. Talk to them. They still have a high fever? Oh, Megan, the rapid testing's coming out this week. So is that the little PCR machine that we talked about, that Cepheid test, or is that Victoria? That's a yes to what, Victoria? The rapid testing? In 15 minutes. Okay, it's the Cepheid test, so it's that rapid PCR, right? Deja, give it 24 hours. Oh, they're getting worse. Deja, I would try calling the COVID. That sounds a lot like flu. Yeah, call the COVID hotline. But it does sound like flu. The vomiting is more common with the flu than it is with COVID. Lots of ginger ale. All right, so the last thing, yeah, flu and no NSAIDs. Don't give them any NSAIDs. So the last thing I wanted to show you guys was an article that actually came to me last night. And it just kind of unnerved me a little bit. It was an interesting one though, and I wanted to share it with you guys, because it is also something else we talked about in our immunology as well as in our viral chapter. So over the weekend, there were 6 tigers at the Bronx Zoo that became COVID-19 infected. So what this means is this virus, in addition to being highly contagious, has the ability to jump the species barrier. So it jumped the species barrier, supposedly, right, from some kind of warm-blooded mammal, bat, to, I forget the name of the mammal, to human. So it jumped the species barrier to human, and now they had blood tests from these tigers that tested positive for COVID-19. They believe they were exposed by a worker from the zoo who also tested positive in New York. This is in the Bronx Zoo. So now we have cats that obviously we now know this virus can replicate and survive in cats. Now the cats were fairly asymptomatic. The symptoms were very mild and it did affect them differently, but they believed to have been infected by some kind of zoo worker. So they do show symptoms, right? They have a dry cough, that sort of thing. And what prompted the veterinarians to test these cats for COVID, I have no idea. But I did look this up in a couple of different resources to make sure that it was real. And the BBC, to me, is a fairly, and CNN and the Washington Post also posted these articles. So I thought those were relatively safe resources. So I thought it was interesting that we have a very rapid shift in crossing that species barrier. That's kind of a big deal in viral infections. Did anybody else see this article? Or see anything about it? Yeah. Were you thinking the same thing? I'm just curious. Were you guys thinking, oh my God, it jumped species? Because you're all nerdy scientists now. That we don't know, right? What came first, the chicken and egg? Yeah. I don't know, Jacqueline, if it means that most of them can. There's a concern on that, right? So if it's in tigers, that means domesticated cats. Cats have a really strange physiology. It's very, very different than, like, say, dogs and even humans. So they are a little unique because they are true carnivores. Their metabolism and their physiology is a little bit different. So Brittany, that sounds awful. Anyway, so there's a concern now if you are infected that perhaps if you have a cat, that the cat could be infected. As far as dogs go, there's been no finding of the virus in dogs. So it's definitely an interesting find. I think they're going to probably start testing cats around the world to see if this is something like looking at people who were infected that have pet cats, have domesticated cats. They may start testing those cats to see if it's something that can be present in the cats as well. So we're going to finish this lecture up on Wednesday. We're done for today. We will finish this lecture up on Wednesday and talk a little bit more about some COVID. We might do a little group project and kind of pop out into groups again and get some data and come back and look at that data as a class. So be prepared for that. It's not going to be anything difficult or crazy, but just we are in the middle of this. We're studying epidemiology and we're in the middle of a pandemic. So I would like to kind of take advantage a little bit of that situation since we're in it and maybe get some learning out of it. Does anybody have any questions for today's lecture? I'm working on case studies. I'm hoping to get those out to you guys this week. I just have to get them assigned. I'm just trying to get them organized. Oh, good, Jessica. I'm glad you found it interesting. It's always, I mean, if we're going to be in this situation, at least let's learn from it and get something out of it. Yeah, I agree. All right, guys, so I'm going to stop the recording here.